Well, I think that all of us are proud of the accomplishments of the cooperative groups, but all of us have been concerned about the slowness of developing new trials, and the NCI actually asked the IOM to carry out a study which would bring together information that's been collecting over the past few years and point out directions for improvement. And I guess the most important data were data that were collected showing that the average clinical trial developed by the cooperative groups took 2 years or more to come to fruition just to begin the trial. That's in designing and getting approvals from all the necessary agencies. And the other data that came out is that if a trial is initiated, designed and completed and initiated from the point of view of getting patients on the trial within a year of its conception, it's very likely that it'll be concluded and accruals will occur and decisions can be made based on the data collected, whereas trials that take 2 years or longer to develop and be launched often aren't completed. So the track record was that approximately 40% of trials that were designed after all this hard work and all this effort and all the participation of patients who were agreeing to participate in the trials in order to not only help themselves but to aid science, 40% of the time we didn't get the answers we were hoping to get because the trial never finished completely. And I think that was a wake-up call to let's streamline this thing, let's get trials out more effectively and efficiently and more quickly and get at it. Well, the NCI is in the leadership and coordinating role, but everybody's going to have to participate. The FDA, the drug companies that produce the drugs, the clinical investigators in academia and in the large group practices in the country, and the NCI are all going to have to, and the patients are all going to have to agree to streamline. And I think the NCI is in the leading role because it puts up about half of the funding that's required to do this, and also has a tremendous infrastructure that allows us to carry out these trials. But we're all going to have to do give and take, and we're all looking at this through different lenses, and we're all going to have to say, well, each person that has their hands on this, each agency that has their hands on this, will have to say, we're not going to keep rewriting these trials and making them more and more perfect. We're going to agree that we'll try to get everything done within less than a year and get the trial started. The problem is that if a trial is devised in academia, maybe 10 different universities have to come to an agreement on the trial, and then the NCI has to come to agreement, then the FDA has to come to agreement, and if there's a question raised, it bounces back, and all 12 universities have to look at it again, and these trials recycle. And if one person's on a ski vacation for a month, that could hold the whole thing up for a month while everybody waits. So the simplest thing would be to lock everybody in a room and say, let's get this done in a week, but that's not practical. And we'd like to find a way to do something in between. Well, the FDA, of course, is in a very challenging position because they're charged to demonstrate efficacy, but also to ensure safety. And balancing those two are very challenging. The FDA is set up with expertise in drugs and separate expertise in devices and other expertises in antibodies and vaccines. And in the case of cancer trials, things are often combined. The trial might have an antibody and a vaccine and a drug all working together in sequence or even at the same time. And the FDA is in a position then with multiple lenses looking at the trial from inside the FDA. We all hope that in the case of cancer, where we're convinced that combined therapies involving 2 or 3 or 4 modalities in a trial are going to be the way to go, it would be more efficient if the FDA had one cancer approval process that could collect all these together. And then the FDA is working hard to develop the proper guidelines that will allow us to combine drugs. And finally, the FDA is working on guidelines to help us decide how to properly use biomarkers to select therapy. And I think that's one of the most important things. In the past, we picked therapy on the basis of previous experience in trials with patients, and then collecting the data, and then moving ahead when an individual patient comes in and saying past experience shows that X, Y, and Z drugs are the best. But today there's more and more emphasis on taking the patient's own tumor and biopsying the tumor again sometimes, and running studies in the tumor to see what its genetic makeup is and what molecules are functioning abnormally in the tumor, and then picking from the more than 800 new drugs that have been devised to target these abnormalities and individualizing, personalizing the therapy for the patient. And the trials that do that involve using these biomarkers, which are, again, tests that are done on the patient's tumor, and then using drugs, and the biomarkers sort of an experiment, and the drugs an experiment, and we need more clear guidelines on how to combine all these innovative new ways of looking at assigning drugs to patients and carrying out trials. Well, a biorepository then is a frozen, a bank which contains frozen tumor specimens that come from patients that have participated in trials. And they're very valuable because the clinical trial gives a result and we find out which patients responded and which patients didn't. We can go back to this frozen bank of tumor specimens and pull out the frozen specimens and study gene abnormalities and molecular abnormalities in these frozen tumors and correlate what we're finding in the molecular and genetic laboratory with what happened to the patients. And we can find out which gene abnormalities might predict a good response to one drug versus another drug, and complications from one drug versus another drug. So it's a huge repository of potential information. And it's worthless unless it's collected properly and stored properly, frozen properly, and the annotation, the clinical data annotation, has to be paired with these specimens, and they have to be kept over weeks and months and even years in safe condition, and then they have to be drawn upon by scientists who are going to ask questions. So it takes research to learn from the repositories, and that takes proposals of experiments, and it takes funding to do the research. And this whole process of setting up and then using biorepositories optimally could be done much more effectively if we work together to systematize things, and then if there's funding to with proper review and vetting of the best funding proposals, research proposals, if there's funding to support the use of the biorepositories.